Good afternoon. Sorry for this delay, but we were also managing some questions regarding the online transmission, but I think now we have the conditions to start. So it is with great pleasure that I welcome you all to this workshop organized within the framework of the European WiseScript project. My name is Fanny Souza. I am Associate Professor at University of Beira Interior and a member of the WiseScript Consortium. And it is a privilege for me to host this event. This workshop, entitled RNA Horizons, an Inspiring Bridge Between Generations, is a cross-disciplinary and inclusive initiative that addresses students and researchers from several scientific fields, including medicine, pharmaceutical sciences, biomedicine, biochemistry, biotechnology. And our goal is to create a space for scientific exchange and discussion that brings together diverse perspectives. To reach the broadest possible audience, we are also broadcasting this event online, enabling participants from all over to join and benefit from today's discussion. We are greatly honored to host two distinguished plenary lecturers, Professor Kathleen Caricot, awarded the 2023 Nobel Prize in Physiology or Medicine, whose groundbreaking research on RNA has paved the way for unprecedented advances in biomedical science, and Professor Chantal Pichon, the principal coordinator of the WiseScript project, whose leadership has been instrumental in driving this European initiative forward. Following these plenary sessions, participants will also have the opportunity to engage in a guide tour on site through the RNA biomanufacturing process for a unique perspective on the science behind the project. We are also honored to have the rector of the University of Beira Interior, Professor Ana Paula Duarte, and the president of the Faculty of Health Science, Professor Miguel Castelo Branco, with us for this opening session. Their support and encouragement have been essential to this event. So I would now like to hand over to the rector of the University of Beira Interior, followed by the president of the Faculty of Health Sciences for their opening remarks.
Hello. Hello, everyone. Good afternoon. Distinguished guests, Nobel laureate Professor Kathleen Caricot, Dr. Chantal Pichon, dear Dean of the Faculty of Health Sciences, dear Professor Fanny, faculty members, researchers, students, and friends. It is a great honor to welcome you all to the University of Beira Interior inside, and online also, like me. For this workshop, I want to express our gratitude to Professor Fanny for organizing and for hosting this event, and also to express our gratitude to the European WiseScript project for this organization. And to Professor Caricot and Dr. Pichon for joining us. Your work inspires us all. Today represents more than just a scientific gathering. The title of this workshop, An Inspiring Bridge Between Generations, reminds us that science advances not only through individual breakthroughs, but also through dialogue, mentorship, and collaboration. It is by connecting experience and wisdom with curiosity and new perspectives that research achieves its most transformative outcomes. For UBI, hosting this workshop aligns deeply with our mission to promote excellence in research, foster international collaboration, and support the development of young scientists who will shape the future of knowledge and innovation. We believe in fostering environments where curiosity thrives, where students and young research are supported, and where complex challenges are met with collaboration and innovation. I hope that every participant finds today's plenary sessions enriching, that you enjoy the RNA Uncovered, an end-to-end scientific tour, and that the conversations sparked here extend far beyond this room into your research, your labs, and your future collaborations. Thank you all for being here. May this workshop open new horizons for us all and strengthen connections between generations and fuel fresh ideas that will advance RNA research and its applications for the benefit of humanity. I extend my warmest welcome to all participants and wish you a productive, stimulating, and memorable workshop. Thank you once again.
Well, I also would like to greet everybody that is present in this session, both by being present in place, and also, and I would like to greet firstly Mrs. Rector and Dr. Professor Fanny Souza também, also, because be very, very grateful for the opportunity of having here this kind of symposium with so important participation by Professor Kathleen Carico and Professor Chantal Pichon. It's for us a real honor to have such a distinguished guests with us today, talking about this so important RNA research and developments that are very important and nowadays, and fortunately for the future also. So, as Mr. Rector has said, it's for us very important to work together interdisciplinary, and our faculty is an example of that. Besides medicine, also as biomedical sciences, pharmaceuticals, and optometrics, and works in the environment in the university with engineering and other disciplines. So it's for us very important to keep this interdisciplinary approach and to be a part of this workshop organized within the scope of the European iScript project. That is very, very interesting, and we're very, very, very interested in the outcomes that could come from this kind of, for one side, science partner, and also talking with such development to be part of the construction of the future within Europe and in the world. So thank you very much for being here and to participate, and I hope this symposium will be a success with the opportunity of talking with such important and such high-level persons. So thank you very much.
Thank you very much for your kind, motivating words, and also for all the support in this organization of the workshop. So now let's move on our first plenary talk. It is a privilege and a huge honor for me to present Professor Katalin Carico, who is presently professor at University of Szeged in Hungary and adjunct professor of neurosurgery at the Perelman School of Medicine in the University of Pennsylvania, where she worked for 24 years. She is a former senior vice president at BioNTech, where she worked between 2013 and 2022. She received her PhD in biochemistry from the University of Szeged in Hungary. For 4 decades, her research has focused on RNA-mediated mechanisms with the ultimate goal of developing in vitro transcribed mRNA for protein therapy. She is a co-inventor of several mRNA-related patents for the application of non-immunogenic modified RNA. Professor Caricot co-founded and until 2013 served as CEO of RNA-RX, a company dedicated to developing nucleoside-modified mRNA for therapy. Her patents, co-invented with Drew Weissman, on nucleoside-modified uridines in mRNA, were used to create the FDA-approved COVID-19 mRNA vaccines by BioNTech, Pfizer, and Moderna to fight the pandemics. For her achievement, she received many prestigious awards, including the Japan Prize, the Horvitz Prize, the Breakthrough Prize, the Lasker-DeBuckett Clinical Medical Research Award, and the 2023 Nobel Prize in Physiology or Medicine. Thanks for your generous participation in this workshop, and we are really excited to hear from you. The screen is yours, Professor Caricot.
Okay, now, so I am here in the United States. Once I was actually in Portugal, and I was in Lisbon in 2015, and I admire all of those styles you have there. So I had so many pictures about that. I thought that maybe I put some of them in the presentation, but rather not. And I am so happy to be here with Chantal Pichon. I met her in France first, and then many times in mRNA therapy meetings. And thank you for Bruno Battista that he invited me. And I am happy to be here and present something about my life and, some message for the young one and about the RNA, so that I will share my screen here. And oops, sorry about that. I don't know that it opened in the very last page, but so quickly I can go through my presentation here. Okay, so this is my presentation here today to talk about developing mRNA for therapy. As I mentioned, I grew up in Hungary, and my parents had just only elementary school education, and we lived in this small adobe house. And actually, that's my sister, but I was just told. And she's 3 years older, and we had a very happy childhood. And my father was butcher, and I learned pig anatomy as a toddler. And so my sister was very good in school, and what it happened usually in this case, that they expected me to be good. I competed in elementary school in geography, chemistry, and so on. And in elementary school, I was third best in the country when I was 13 years old. And so after that, I went to the high school, the local high school, and where we sent letter to famous Hungarians, because we just could write in Hungarian. I learned Russian and Latin in this high school. And why I am mentioning, because we sent a letter to Shaya, who was investigating stress. And he responded, and we were so excited, and we read his book. I have to tell you that I wouldn't be here. I wouldn't get any award, any of these things. I would not learn at age 16 how to handle stress. I read his book, Stress of Life. Of course, it was reading in Hungarian. I couldn't speak any English until I went to the university. So it is very important for you, for your mental health, to learn how to handle the stress. you always feel that your colleagues are advancing and you are not. And if you are pay attention on this one, you already took away your attention what you can do, because you cannot influence that one. And this is like a Stoic philosophy, that you always focus on things what you can do and not what others should do. And he demonstrated that the stress can kill you, but only how you perceive it. And so if you learn to perceive properly, then you can be much happier, even the same circumstances what you have right now. And in my book, I also quoted many things which I took from this book, and how I can survive the criticism, the hardship, and all of these things, and come out with a happy life. Anyway, in the small town, that my goal was to go to the university, and I want to go to University of Szeged because they just in the same year, in 73, they opened this biological research center, and that was my dream. I will be a scientist in white coat. I will be standing right next to a bench and then discovering something like a vitamin or something. I don't know what I had in mind. So when I was student, I already went to this institution. It belonged to the academy, and then I tried to find a place. And I was interested in plants. Most of the people were interested in genetics, but the only opening was in the lipid team. So I thought that the first summer I would spend there in the bench, but the lipid team had told me that go to field trip. And so I eventually ended up in collecting a sample from fish, which was farm fish, and collecting water fleas. So what I am saying to you, that you have an imagination, something about your future. But whatever life throw in you, you just have to take it seriously and learn from it. And that's what I did. And actually, my first paper, I was participating in these studies, came out from this field trip. And when I went back in the fall to the same team, there were two scientists who were, interested to make liposome. Of course, I was student, I never heard about liposome, but, they wanted to have this phosphatidylserine ethanolamine mixture, and they came to the lipid team to have them. And we isolated this from a co-brain according to a 1942 publication, because I was behind the Iron Curtain. This product was available in Germany, but it was under embargo, and so it came handy that my father was a butcher, and I was not ghoulish about working with co-brain and isolating lipids, and that's what we did, and delivered this DNA, plasmid DNA, to cells using this liposome. And one day, walk in Professor Thomas Jenny, and to the lab, and he told me that he is heading the RNA lab and whether I want to make my PhD there. So you can see that I was not this visionary person who figured out, okay, now I know liposome and delivering nucleic acid. Now I have to learn more about RNA, how to synthesize, how to test them. It was just I went with the flow. And he told me about this oligoadenylate that I have to synthesize, this short RNA molecule, and he asked me to set up a viral screening lab. And I worked together with a fellow graduate student, János Ludwig, who even today I work together. We have patent and publication together. So it is a message to you that be nice to your fellow graduate students, because you need their expertise. Everybody around me was actually organic chemist, and I was the biologist set up for screening for this molecule. This molecule is natural. Oligoadenylate synthase enzyme makes it in response to double-stranded RNA. And when this molecule, this small RNA molecule is made, it helps to dimerize latent RNAs, which will degrade the RNA. And that's what the idea, that the viral replication is inhibited. So everything kind of look good in my life, things going well, setting up everything, but all of a sudden the support from the pharmaceutical industry ended, and so I Meantime, I had a husband and a daughter, and we had to leave. I lost my job in Hungary and ended up in the United States. I seem very happy there, but I have to say that I never wanted to go there. I was so happy in Hungary, but that was the only choice I could have. And so I started to work in Professor Suhadolnik's lab, and we did also oligoadenylates, different modified. We tried to develop antiviral compound because it's badly needed. The problem was there in 1985, we are talking about, is HIV. But eventually, we couldn't make this small 2,5-A molecule to work, and we synthesized long double-stranded RNA, which was known to activate a system and generate interferon, but it was toxic, so we made a mismatched double-stranded RNA, and we did a human clinical trial. We treated HIV patient in 86. Unfortunately, there are system is compromised, interferon system, and we couldn't have them. So up until that, now I am 30-something years old, and I did the small RNA, I did long RNA, double-stranded RNA, but not mRNA yet. So the messenger RNA was discovered in 1961, and every 20 years something happened when eventually 60 years later it was an FDA product in the form of mRNA COVID-19 vaccine. So the first 20 years scientists just could isolate RNA and analyze them, and from 1984 when they discovered how they can make in a test tube, then you can see that studies started in animals and try to figure out what it could be useful for. 2000 was the first RNA company was formed, and then clinical trial started in that. So this was very important to know how to make in a tube. And in the early 90s, more and more scientists started to work with the messenger RNA, and especially when John Wolf and team showed that when they injected in animals, the RNA translate and detect the protein. And every year, there is a publication out. So for me, it was easy to follow the literature. And first, therapeutic use of mRNA was reported, and that was what I always wanted. So even today, I wanted to use the messenger RNA for therapeutics. And in Floyd Bloom, who was at that time was the editor of Science, and he was at the Scripps, so that they demonstrated that injecting vasopressin in the rat brain was treating a disease. And following years where influenza vaccine were developed, even self-amplifying RNA was used, cancer vaccine. Except that, 1996, Eli Gilboa, they demonstrated, the mRNA can be used as a vaccine, and they did a human trial all the way to phase 3. But, it was ex vivo treated dendritic cells, but somehow that was not helpful. And when I was following the field, I found that these scientists like Floyd Bloom, they never published another mRNA paper, and scientists from France, they did not publish again. And one reason could be that the RNA was translated poorly, very quickly degraded, and very small amount of protein was produced for a very short period of time. Of course, just recently when I traveled and met some of them, I learned that they couldn't get grant. But I didn't know at that time. I started to work at the University of Pennsylvania in 1989, and that's when I started to work with mRNA. Again, from Temple University, I was terminated. So it many times happened in my life, and I wouldn't be here if I wouldn't be kicked out that many times in my life. So it depends on how I perceive. I never took it, that people said a bad thing about me. Anyway, so with Elliot, he was interested in urokinase receptor, and then I generated the mRNA, and for the functional receptor needed post-translation modification, glycosylation, GPI anchor, so on. And voila, the cell was so smart, knew how to do all of this and generated a functional protein. So at that point, I knew that this mRNA will be useful for something, and I just proceed. But again, I lost my job. I was also demoted from the faculty position at Penn. But luckily, David Langer, who was a medical student in Eliot's lab, by that time, became a resident, and he convinced the chairman to give me a space at the neurosurgery department. So I did all of my RNA work in this small lab here for 17 years. And the budget was low, no lab coat, other things. But I was very enthusiastic. And with David, we tried to develop mRNA for therapy. And so it was, I was writing grants and so on. I never get one. And now that actually, what was the reason? Now people are analyzing why I never get money, and even a problem is named after me. They said that the insider who had the prestige, the connection, the money, the influence, the power, they have everything. And I was an outsider. They said that I just have rejection and disrespect, but I had the freedom to investigate whatever I wanted. So that was mRNA, but only if I have connection to the middle who had the money. And that was Eliot, and that was David who helped me. What happened is that how I see it, that you are doing research because you love it, and you get some result and you want to tell the people that what you discovered, and so you have a publication, and then you need a following to do more research, and you write the grants. If you are lucky, then you get the money. For me, the circle was shortcut here and back to research because I never get the money. But if you get the money, then you hire more people. Students are coming. Oh, they have to graduate. So now you start to publishing even when things are not ready. But the leader of the team needs promotion. Everybody needs promotion. And somehow the goal is now became, more publication, more things to do is for the advancing the career, not learning more about nature. So what was said that the goal of research is changed. The 19th century scientists want to understand nature. You, the 21st century scientists, were published in Nature. So that's the major problem here. So anyway, I couldn't get money, but neurosurgery department supported me. And one day, at the Xerox machine in 1987, I met 1997, sorry, Drew Weissman. By that point, I was working almost 10 years on mRNA, and he was interested to develop it to vaccine. And when he did the experiment, we measured, I made the RNA, and then we found that it was inducing TNF-alpha, so it was inflammatory. We didn't know prior to that the RNA is inflammatory, so we want to figure out that all of the RNA cause inflammation if we expose it to human dendritic cells. And since I was working so many different RNA, I already had a repertoire of RNA in the freezer, and we tested them out. And what was surprising is that when I made the in vitro, the transcribed RNA, that it was very inflammatory, but these other isolated RNA were less inflammatory, and most importantly, tRNA was not inflammatory at all. Couldn't cause any TNF-alpha. And of course, we knew that tRNA has a lot of modification. 25% of the nucleosides are modified. And so that came the idea that could it be that if modification makes the RNA non-inflammatory? So, okay, we should make and prove it, that the RNA is has to be modified, but which one the modification? There are more than 100 modifications are present in the RNA, and which one we should select? And the enzymes which introducing is not even known, so I couldn't call up some company to send me an enzyme to modify. So eventually, what we could do is that we purchased nucleoside triphosphate, which were already modified, and try to see that whether T7 RNA polymerase incorporate when we are generating mRNA in the tube. And some of them incorporated, some of them for obvious reason not. But we could have these RNA, and then we could test them that whether they are less inflammatory. And when we isolated human dendritic cells and exposed to these RNA, we found that some RNA was as inflammatory even after modification, like the RNA which I generated in the cube, which had absolutely no modification, but some were not inflammatory. And when we made a closer look, we found that those when the uridine was modified to either pseudouridine or 5-methyluridine, 2-thiouridine, then those were not inflammatory. So who knew that uridine makes the RNA inflammatory? And then when we modify it, it is not inflammatory. But of course, Drew wanted to make a vaccine. I want to make a therapeutic coding mRNA and therapeutic protein coding RNA. So it was important that it is translatable. And that was our biggest surprise, that the pseudouridine containing RNA, when it translated, we get 10 times more protein. So it was a total happiness now that I can have an mRNA which makes more protein, can generate more protein, and then it is not inflammatory. So what happened here is that we wrote a patent and then established a company. It is very important. The patent is not sufficient. So nobody will go to the University of Pennsylvania patent office and say, oh, pseudouridine, that's a great idea. You know the most that what you are discovering is what could be useful for, and so you have to make a company. In Europe, 83% of the patent is not used. So you have to make the courage and leave the academic position and then push and prove that your product is helpful. And then we wrote a grant, and that was the only one in my life I ever get. This was a small business grant. We get almost $1 million for showing that if we make nucleoside modified RNA, it is useful to treatment anemia. So what we did, we really generated mRNA, very small amount, 0.1 microgram pseudouridine containing erythropoietin RNA. When we injected to the mice, we could detect even 4 days later. I have to say that the erythropoietin half-life is about 2 hours. So when we injected the protein, it quickly disappeared, or when the RNA had no modification, we also could detect short period of time. The generated protein from this RNA was functional because we had high hematocrit and weekly injection of this mRNA, very small amount, 0.1 microgram, sufficient to maintain high hematocrit value. And luckily, of course, the pseudouridine containing RNA was not inflammatory. So everything seems so great. 2012. My daughter get another Olympic gold medal in London because meantime she grew up, and in Beijing she get a gold medal in the US Women 8, and London 2012 she get another gold medal. But now that the University of Pennsylvania thought that I don't get enough money and don't get grants, so I was terminated in my position and forced to retire. Again, this is somebody's decision. I could sit at home and watch the grass growing, but I felt that I have more to offer. So what happened is I moved to Germany and left my family behind and started to work. And not this nice building here now that it was not open. We work on the campus and with Islam and Ugo Zahin, who established BioNTech. And we did further improvement and wrote, a nice review about what the mRNA could be useful for. And so as a result of all of this work, codon optimization, optimizing untranslated region, purification, and so on, eventually, and thanks to other work, this small amount of protein, short period of time, now we could have significant amount of protein when we did all of these changes, and it is extended time. So the RNA was ready for prime time. I have to say that it is not just happening what we improved on RNA, but in many levels. So if I look at there, the DNA template, what we use for mRNA synthesis, I remember when sending out letters to colleagues who cloned certain genes to send me the genes. But after 1989, I started to use for template RT-PCR. This was a TA cloning. So if I had a cell line, I could generate the gene and template for mRNA. Then we started to order clones from Origin and others. And it's about 2006 when custom gene synthesis was available. It was in Germany, there was two companies open in 2000, but for synthesizing genes like GeneArt and Telecon, but the price was affordable about from 2006. So now that it's simplified how to somebody can make mRNA. Of course, the translation was improved. When the cap was discovered, pharmacia started to selling cap. Later, we could purchase polymerase and capping enzyme and so on. And eventually, we could also get clean cap. We could modify the RNA. So translation improved by all of these invention. And of course, the most important is, of course, the delivery. So as I mentioned at the beginning, we also use liposome, and later we use lipofact in transit, which was commercially available product. But for clinical trial, use protamine and later on LMP. So all of these improvement on different level is help to advance our field. And of course, why I'm talking about this importance of this mRNA as a therapeutic, because so many product with protein, which is FDA approved and different things is protein by nature, but it is very expensive. And if we would, instead of using recombinant protein, we could use this mRNA and let the body make the protein, it would be, the therapeutic protein would be much cheaper. It would accelerate the screening, and it would be properly folded, and you don't have to worry about complex purification procedure. So this is how our field advanced, and the timeline that 2020, when all of a sudden people learned the word mRNA, January, we learned the information about the SARS-CoV-2, and Moderna already started clinical trial in March. And by the end of that year, we could get a vaccine. And of course, many people, were worried about that it was so fast, but you could see that there were like 60 years of work went into it. And so this was too quickly developed. But I have to say that those people who are worried that it was the first time we used the mRNA in this vaccine, I have to say that CureVac already started a clinical trial for mRNA-based vaccines for treating or preventing rabies infection, and Moderna had several clinical trials for avian flu, Zika, and CMV, and this was nucleoside-modified RNA with LMP. And so these were clinical human trials, but the people did not know about. So what happened is now that we have more than 150 mRNA-based clinical trial ongoing. Half of them is for infectious diseases, mostly viral diseases, that influenza, RSV. For RSV, actually, one is already FDA approved for Moderna, and there are herpes simplex, CMV, Nipah, norovirus, monkeypox, and again, bacteria like TB, there is a clinical trial ongoing, or Borrelia that caused the Lyme disease. There are clinical trial, and recently Sanofi started acne preventing bacterial vaccine. And for malaria, the vaccine is also in clinical trial. For cancer, that's mostly in what you could see in the news, mostly shared fixed cancer antigens. They are targeting, but there are individualized vaccines which are very successful. And there are also mRNA used as coding for cytokines, so it is not all of them is vaccine, and using CAR T cells and mostly delivering for in vivo now. And before 2020, cardiovascular diseases were in the focus, and this already phase 2 trial was before 2020, VEGF-A mRNA for heart failure. And recently, more genetic diseases targeting with mRNA, mRNA coding for the missing proteins. And autoimmune disease like this using CAR T cells, and for myasthenia gravis already phase 3 trial, or lupus in phase 1 trial. So that there are more and more mRNA therapy is ongoing, although you are probably aware of that in the United States that mRNA became a dirty word. But if you look for clinical trials, you will see that many of them is running in China. So what is left for me is, inspiring the next gen. Oh, there is still, want to mention that mRNA is also helping animals, like the zoo animals. Here in Texas, they used vaccination for elephants. This was a little bit higher dose, but 150 microgram RNA was used, or horses, and in France they used in dogs. And for me is inspiring the next generation. These are books for children that was wrote about me. I didn't wrote these books. But, here is Drew Weissman and me working in the lab, and, everywhere you can see the glass tubes. But I have to say that I have not seen a glass test tube when I was student, maybe. No. But I also wrote a book about my life. And this is out in different languages, and not in Portuguese, but it will be published in Portuguese in Brazil. But so Spanish and English and so many other languages. And I try to describe my decision making and my life, and how, what I mentioned in the beginning, how important is to believing in yourself, to mentally, physically health. I have to say that I was running every morning before work, and even in Germany, 6 kilometers before I went to the lab. And now I'm swimming or doing some activity. You have to do it. You have to be healthy, and then you can change the world, and then you can do all of your research. You have to believe in yourself, mental and physical health, and the rest of it is just fun. So with that, I just want to finish and stop here sharing. Thank you for your attention.
Thank you, Professor Caricó. Thank you so much for the insightful and the fascinating talk with the different perspectives for the scientific experience, and also the personal perspective, and also the advices to the youngers and the young researchers. It was really interesting. So I will now open the session for the questions of the audience. For those who are attending online, please raise your hand or put the question in the chat, and we will try to transfer and put the question. Chantal.
Thank you so much. Katie, I'm so grateful that you accepted to do this. As you know, I just want to say that it was a wonderful talk and inspiring. I just want to, for the Y script, this is our last workshop, and I'm so glad that actually Bruno, one of young PhD student, is taking the everything, and he started to tell us we are going to invite Professor Caricó, and I said, oh my God, she has a lot of things to do. I don't know. So I think it's very inspiring. I just want to say, and to just to share with all of the audience that actually, if I'm still, doing this mRNA work, I just want to say that Katy Carico was behind me. Of course, I also asked for a grant. I didn't get it. And then, I was just want to stop it. And I remember a word, Katy, and I would never forget. You just told me, Chantal, keep going, work hard, and think outside the box. I would never forget this. And you know what? I think it did work, right? Because I really work very, very hard. And that's why WASCRIPT actually, is here. And we are working and still working and have a great, partner as a Fanny. So thank you so much. This is just a few words for you. And just to tell all of the young people listening, there are many PhD students of mine actually listening. Yeah, they're just waiting for your talk for a long time. So I think, yes, I think it's really great. And I think, yes, I just also take one of your words, actually, one of them you put in your slide. Don't try to look successfully. I think it's so, so, so true. Thank you so much, Katy. Thank you. And that's it. Thank you.
More questions? I think we have a question here in the room.
So thank you. First of all, thank you very much for your very inspiring talk. And my question is about the delivery systems. Do you think that delivery methods behind lipid nanoparticles will be needed to reach certain disease?
Oh, there are many different formulations, not only lipid, but other polymers are used. And the most advanced is that they try to target specific cells, cell types, bone marrow. So then many of the therapy, which is now requiring taking of the bone marrow and very expensive, targeting it directly. So just delivering the mRNA in certain ways to formulate it can reach the bone marrow and then can change it, and then the treatment could be much cheaper and faster. And so I don't know whether this was the question, that whether this, the future is about 99% of the papers is now that how to target and how to deliver and making simpler the technology. So is it that what you want to know?
Yes, thank you. Thank you very much.
Yeah.
More questions?
So where you are thinking about the question, I can say, that I mentioned that I have a daughter, so that I want to say to those girls that you don't have to choose between career and the family. I didn't overdote in my daughter, that she just watched me working and my husband working hard, and then the child will watch it, and then she came out okay. She get an MBA, and now she's heading a company we established, and so it is. And then she get 5 times world champion, 2 gold medal in the Olympics. What more you want? And so that. That's very, very important. And so for that one, you have to find the right man, the husband who appreciate that you have dreams. And guys there in the audience also who is watching, be a good husband, that realize that your wife has dreams and help, because that's important. Without my husband, again, I wouldn't be here. And again, not to look successful. When I met my husband and married, he worked in a factory. And not a lawyer or somebody who's doctors or somebody, so that's not for success. Nobody would say, oh my God, great marriage. For me it was. And that's what you have to make your own decision, which is good for you, and not for somebody show up that you are so successful.
Great advice. And we always find balance between the things. Yeah. I also kept your words to be honest with what we are doing.
And don't try to find a good work and home life balance. Yeah, it is very difficult to be a scientist and have this balance, but whatever you find, it will be fine. Okay.
Maybe I can also do with a question, a curiosity, because you were mainly working with RNA when the majority of the persons were working with DNA for gene therapy. So what was your motivation? How do you followed with the idea of working with RNA? What inspired you in the RNA and not so much in DNA?
Yeah, it is. When I said to people that I work with the mRNA, they felt sorry. Poor Kati, my God, I hate RNA. It always degrades and so on. So that, they felt sorry for me. But because, at the beginning, I already in Hungary, I work with RNA. I know the enzyme, how to label, how to modify, how to generate. And you have this vast amount of knowledge. You have this knowledge, and then I could see the progress. So as not I didn't give up because I said shouldn't give up because I could see the progress. So when I could get the enzyme and get poly-A tail on it, I could see, oh, much better. And then when I figure out certain formulation, oh, well, okay, it is even more protein. And now I had to figure out what kind of disease is I can treat with that. So try to find, the code for, now I had the button there. And so it was constant, and the purification gave like 2 log or more protein. So every time I figured out that something is better, and people who talk to me down, they didn't see the result, although I published, but they not in the top journals. So actually the Nobel Prize was given, and they named 3 papers, and Science and Nature was none, because I did not publish there. But it's okay. Nucleic acid research is a good paper, or so that they are happy that that was quoted. So that now that everybody can read online, it doesn't matter which journal is published, really.
Thank you. Some questions? I think we have another one in the room.
Can you hear me? Yeah. So I have a question. It's not a scientific question. It's more a personal question. What changed in your life, in your research, when you win the Nobel Prize?
So I have to say that 3 years before the Nobel Prize, in 2021, I started to get awards. And maybe you can see behind me, there is unbelievable. My life immediately changed. I was immediately pushed to the spotlight, and then I had to give interviews. And I mean, I was a scientist in like pipetting and quiet, and nobody invited me to talk anywhere, and all of a sudden is this attention. And I had to realize that I am representing you, all of the scientists. And I always, whenever I thanks to all of the people, that who came before us and all. So I had to realize that is my responsibility, to talk to all of the scientists who I was for 40 years, never get a grant, never get any award or any kind of acknowledgement. I representing all of them, all of you, and that was one thing. And also had to realize that, especially that the people against the scientists, we, is somehow that we are not speaking to the public. And that's why United States get where we are now, because we did not talk. So as a Stoic, I look for what we can do. So we have to go out and talk to the public about what we are doing. And we are not an elite. We are one of them. And that's, realization. And of course, Nobel Prize is so well known, and people said that everybody's dreaming about. I have to say that I never dream about any kind of award. Those are given our people, that some committee people in Karolinska right now, they are deciding who will be next week, or after that. So it is not depends on you. You have to always set up a goal that depends on you. You study and better understand the process. avoid somebody else's decision. You cannot influence that. So, yeah, I don't know. Of course, it's recognition that if I go somewhere and people recognize me, but when I met Bill Gates I was Time 100 most important, and I don't have to say who I am. He knows, oh, I am so happy to see you. He knew who I am. So that's sometimes it surprised me that, but I could live without all of this recognition. That was the other things, I didn't crave recognition. When I organize RNA therapy meeting, I always push forward the young one to present, and I was not putting myself in the front. So that's, yeah.
Thank you. We also have a question from the chat, which is from Bruno, who was always in contact with you, but he is in the technical part, controlling the online session. So he put the question on chat. So behind vaccines, which medical areas do you believe mRNA will have the biggest impact next?
Yes, so many fields. What I mentioned, the clinical trial ongoing on this vaccine is so quickly, the importance is so quickly can be generated a vaccine. And so is definitely is helping, the emerging diseases to treat, and with different viral diseases in Africa and other part of the world, and of course the cancer vaccine, so that better understanding the immune system will help us further that, and and autoimmune disease, that's somewhere there is no other solution. So mRNA therapy meeting will be in November, and the emphasis will be on autoimmune diseases where mRNA is used. And so I am rooting for all of the field and helping as much as I can.
Thank you. And also from Jeremy Burato, I think, to you, what are the biggest challenges to have even better therapeutic messenger RNA?
So, there are still immune responses, and so that maybe Chantal will talk about that.
Yeah, I think from Chantal, it is the process.
To make it simpler.
Production and
better.
purification.
Yes, yes. So that it is up to the audience here, they can come up with things, new idea, new approach. Just like, when you have a candle and improving more light, not automatically is coming to get a light bulb. So it is, different thinking is needed, and that the young one there, I am sure that they will come up with some new things.
I think Chantal also wants to add something. Yeah.
Okay. Yeah, Katie, just want to ask you, if you look at the PubMed every week, we have thousands of papers, 2,000 of paper on mRNA, and my heart is beating every time, because I oh my God, someone is going to. How do you think about how quicker, how fast, how this field is growing up these last years? And if you look at all of those papers from all over the world, I'm just, sometimes, and if you look deeply, I remember you used to tell me, you just have to read thoroughly and look and see. And if you read thoroughly, and sometimes in an amazing paper, Nature, PWS, Science, whatever, you think like, okay, I'm gonna make this in my lab, and most of the time, I have to say, sometimes it's not working. So I just want to have your opinion on, do you really think that all of those huge amount of information that we got from, sometimes I'm thinking like, are we going to be overwhelmed with all of those things? We don't have enough time to really process, to really, as you said, learn as a scientist. I really want to have your opinion on that.
So for me, because now I'm 70 years old, so it was easy because at the beginning the paper were just 4 or 5 pages. And if I read all paper, I love to read like 50s and 60s. I can see that less data, but more thinking. What the scientists were thinking. I was surprised that they already had a conclusion, which we just later proved, so that more of the thinking, and you can think together with the scientists. And now the pages are 35 page of, in Nature when we want to read, and it is, difficult to understand or immediately the relevance of to your work. And yesterday I was just reading one with the telomere extension and said, oh yeah, and 20 years ago I spent with the Christmas figuring out, and I made telomere, and just recognizing, oh, I did that, or reading about the circular RNA. And in 1993, I had a rejected paper on circular RNA when people are saying everything is new. So I just, some critical reading that, when I read a paper and I can see a misstatement or somebody did not read, I stop reading because I said, that they have not done the work. And they sometimes just see that how they evaluate something or what they did, and quickly I can select out that is not relevant or not reliable. But I know that for the young one it's difficult because they would spend so much time reading, and they don't have that eye for it, that okay, um, if they quote a paper which was already, I know that it was not proven, like Rigai was activated by triphosphate RNA in 2006, and I can see that they are still referring. I know that they are not up to date, and I stop. And but it is difficult to be selective and make sure that you don't miss the important ones. So that's why the network of scientists is important. So, with my new colleagues, I send sometimes that, did you see that? You don't miss that. And so that in this way, you don't miss important papers if you have a network. So you have to have colleagues and fellow scientists, and sending each other something that this is important, you should look at. So that's what the way you can select.
Yeah, I think you are right. I think this is a very wise word for all of the young people, because I think sometimes it's so difficult. And sometimes, you're a little bit, I would say, okay, maybe sometimes you do not have enough strength, enough work, force to do everything in a quicker time. And I want to say this to all of the students that are listening to us. But we have to, of course, there are a lot of things is going on. But if you are doing the step by step what we are doing, I think you still have some place to publish your work, even though it's not going to be in Nature, because many students are actually working like, we need to have a Nature paper.
Oh, I showed you. That's the 21st science.
Yeah, that's why it's really I really taking this time with you to tell the people.
I have to say, Chantal, actually, that when I was reading something, I always look for, because you remember something to read when you are emotionally excited, and I want to know, did they found this? And then I'm so high up on that, and that's why. So I never read that, okay, I should read that. The third sentence, I would forget, what am I doing? What did I write? It's nothing. So that I always like Columbo, looking for the clues, that did they
Searching, yeah.
or look at the, the technique, what did they use? Because I did I was 58 years old. I did all of the experiment myself with my hands. I poured the gel, cultured the bacteria, isolated the plasmid, linearized, checking out the sequences, everything I did. So had a good experience on it. And when I look at the technique, I can say, okay, that's not reliable. I know that.
Yeah,
But
thank
you
you.
have to have something to look for, so that I have sometimes idea very far away things, and then I was looking for something and finding a paper, in that paper a reference, and finally it is already in midnight, and I'm still so high on. Somebody must measure this already, and I've had this feeling.
Yeah,
I try
yeah,
to find it. So
yeah.
excitement is needed. If you
Yes,
just read the paper because the
you
supervisor
have to
told
read.
me to
It's
read
not.
it, then
No,
third sentence you
I
you
agree.
remember.
I agree. I agree. I completely agree. But in the delivery systems, Kati, there are a lot of papers, great papers, I have to say, but a lot of them. Thank you so much.
Thank you. I think we have still space for one question from also from online, Tanya Hamun. Well, she did first a comment, really inspiring presentation, and then the question, do you think that using modified uridine in siRNA would be interesting for siRNA therapy?
I mean, the siRNA, they deliver in double stranded, and it is the stability is there. I am sure now that with the other RNA, they tested out microRNA, other, introduce modification, or the guide RNA, for example, for others. So, people are trying out things, and see that whether something is better. But um, you have to know that the pseudouridine is the fifth most abundant nucleoside, and when we discovered that we were putting mRNA into our mRNA, it was not known. It was 10 years later, 2014 or something, when they discovered that actually messenger RNA does have pseudouridine in it, and so I don't know whether to helpful or not, but it is a different issue because those are double-stranded RNA and they are stable, and GalNAc targeted in the blood is not degrading, so the stability is not as a serious issue.
One comment also online from Eric. He makes a citation from Mark Twain. They didn't know it was impossible, so they did it. It was not, of course, about mRNAs, but it could have been. And congratulations for your career.
Yeah,
And
actually, I used to say to when they ask me, about whether I think something is possible, I said I wouldn't say. I would be the last one to say that. Oh no, don't try.
Because you did it. Yes. Yes. Yeah.
one last question here in the room.
Hello, can you hear me?
Yes.
So my question is kind of in two parts. So first of all, you talked a lot about mRNA and the problems with it, about cytotoxicity and immune reaction, and also its proneness to get to be degraded by RNases and so on. So my first question in the first part is, what's your opinion on the integration of that so-said RNA strands in a more complex system, such as circular RNAs that can bolster more types of mRNAs in the same molecule? And as you said, mRNA, that's the second part of the question, mRNA research took 60 years until the first FDA approved technology. And with the crosstalking technology today, would it take another 60 years for said technology, such as circRNAs, to get on point and to be used and to be approved?
Oh, so circular RNA, you have to understand that when the people tested out circular RNA and so that demonstrated the superior translation, those were done in a human maintained cell line. And if you know that how you can generate a maintain permanent cell line is that you knock out T2 RNAs. So they don't have the T2 RNAs. And then when they tested out in mice that how well the circular RNA work, then you have to know that they don't have toll-like receptor 8. So that it has to be human cells to test out. And so there are sometimes showed advantages like the replicative RNA, which work very well in mice. But because, when you have replication, then you need more nucleoside, and only cells that dividing has plenty of nucleosides. And when it eventually went to larger animals, it was the same amount was required from the replicative RNA as from the conventional RNA. So that, you have to always think what your system is, what you are measuring. And so I don't know. They simplified the circular RNA. They showed that it can be capped RNA. It can be even trans to put the cap there, so it's not a iris element is needed to incorporate. So that there are progress is there, and maybe selectively is good, because maybe for cancer cells, because they don't have the T2 RNAs and they won't degrade, so it may be selective. And you have to think how to make from the lemon a lemonade, that you using some limitation of the technology to benefit of the patient. And so it is I don't think that it will take that much time again, that but how we know the future. I mean, when I started to work, listen, there was no PubMed. We just talked. There were no PubMed. There were no gene bank. Gene bank was in 1990 or something. And PubMed also, and we couldn't listen a lecture because YouTube was from 2005. So things when I was doing, we had no computer, and then so it's just things happening, and then what you have to learn is to adjust to the changing conditions. And what remember that one thing which is for me and you is true: we just have 24 hours. So make sure that you are not distracted on other things, and use your time wisely. You didn't ask for that, but okay.
Thank you. And probably I would ask if you can give an advice, a final advice, or what you could say to young researchers, to the students who want to follow, to invest their time in a project that could be a risky project. So what can you say to them?
Yeah, so that you are young and you take the risk, and you might be in academia, go to the industry. I mean, I never mentioned that, but going to biotech was very refreshing for me, because finally it's not how many reference, how many paper. I can tell you guys, I had not even published 100 papers, because it is difficult when you are doing all of the experiment writing up. You won't get a year 100 papers, in lifetime you don't get. So it is this difficulties of yeah, I don't even know what we were advice. Okay, so that so you have to think about writing a patent if you discover something, as I mentioned, and spin out a company. I know that it in Europe a little bit more difficult to get funding, even now that here is also difficult. But you just have to be brave and take the risk, because when you are young, you can more easily can do that. I was old and I could do, go somewhere. But you realize that if you go to a company, if it is a big company, you are a little something you are doing, but it is up to you. You can learn so many things. You go to a biotech company, like I went, you have to realize, okay, every day what you are doing can impact that, whether that the company will be there tomorrow. But there, Ugur had hundreds of ideas, and I went there, 30 ideas, and zero was made from it, because it was his company. You have to realize you have to make your own company to do what you want. And that's what I am doing right now. Now, finally, I get it. So that you take the risk. That's what. And then you know, you can work day and night and work very hard. But if you believe what you are doing is good, then that's it. So it is good to go out, go another country, go somewhere else, because not only you learn about that other culture, how people are doing in other place, you learn more about yourself. And you realize, oh my God, I can do that. And you did not know. So you discover more about yourself than even about the new environment. And so that's what I would suggest to the young one. Be brave, go somewhere. And listen, I never had famous advisor, and that's what is, you know, they look at me always, you know, coming from a who knows where is Hungary. Never heard about university and no famous sponsor or advisor. And it seemed like, you know, I was nobody. But I never took myself that I am that. I always knew that I have the knowledge, and that's what you have to build, this self-belief that hard work, I can reach my goals.
Thank you so much. Thank you for your participation, for your generous opinion and sharing all the experience and inspiring talk. And I also have to leave a special thanks to Bruno. Chantal already mentioned that he challenged me. So he is my PhD student. He challenged me. He always believed that we could have a talk from Professor Carico. So it is great also to believe in our students and follow their opinions. Thank you so much.
Thank you.
Thank you. Thank you so much, Kathy. Thank you.
Thank you. And now it is the moment to present Professor Chantal Pichon. So I'm also very grateful to Professor Chantal for joining us at this workshop. I've known Professor Chantal for over more than 15 years, I believe, and we have been collaborating in several projects. So it's always a privilege to hear from you, Chantal, and learn. So Professor Chantal Pichon currently serves as a full professor at the University of Orléans in France and is the director of ART-ARNM, an R&D lab of INSERM, where she leads national efforts in mRNA therapeutic innovation. She holds a chair of innovation on biology and health at the Institut Universitaire de France, a distinction reserved for the top 2% of professors in the country. A globally recognized expert in nucleic acid therapeutics, Professor Pichon has made pioneering contributions to mRNA vaccines and protein replacement. She developed original mRNA lipid-based delivery systems, and recently she patented yeast-based system for scalable, cost-effective mRNA production, making a major shift in the field. Professor Pichon currently chairs the Infectious Disease and Vaccine Committee of the European Society of Cell and Gene Therapy. She's a board member of the French Society of Nanomedicine and serves an expert in different private companies to bring strategic insights and translation expertise in RNA-based medicine. As director of the INSERM ARNM Accelerated, she spearheads the integration of cutting-edge mRNA strategies into biomedical and industrial applications. Thank you, Chantal, and the screen is yours.
Thank you so much. Thank you so much, Fanny. As you said, we've known each other for a long time. We've supervised 3/4 PhD student, and Bruno is one of them. And I'm so glad. I would like to thank the Faculty of Science, the Professor Ana Duarte, and also Professor Castelbranco. I mean, when I'm coming to University of Sevilla, I'm always very well welcomed. So thank you so much. Yeah, when I just want to, I already said thank you to Katie, because I think for me it's really very important to have her for this last workshop of YScript, because when Katie Carico was the first scientist I talk about this YScript. I just want to tell you. And yeah, and she didn't tell me, oh, this is silly. She didn't tell me that. She just say, go on. And she really was very supportive, and thank you so much. I'm going to share my screen. And of course, what I'm going to do today is to give you where we are. Of course, I cannot tell everything, as you can imagine. But I think it's important to give a kind of where we are, what was the idea at the beginning and where we are now. And hopefully, I hope that in 2-3 years we're going to have something interesting. So you know that mRNA is already known as a disruptive drug today for both
Sorry,
personalized.
Chantal.
Yeah.
Chantal, sorry, because we are seeing the screen with the notes. We are not in the presentation mode.
Really? Okay. Okay, let me just try to do it. What happened? Oh, I have two screens. That's why. No? Good.
No, we can see now.
Thank you.
Anything.
Oh, sorry.
Yeah, it's perfect. Perfect now.
I have two screens. That's right.
Yeah, perfect.
So yeah, so as I said, you can use mRNA for personalized and universal therapeutic strategy, as Professor Carico said just before. But you know, there is all of this innovative therapy, there is a huge social disparities between and within European countries and all over the world. This is because we have a negatively, you have an association because it's negatively associated, with the access to new biopharmaceuticals. It's very expensive. So I used to say that I'm coming, even though I am French, I'm coming from Madagascar, which is one of a very poor country in the world. And as such, I'm always, you know, thinking about how can everything that I am trying to do, I try to make it in I tried, as I said, to make it in with a low cost. So we really need to have a mitigation action for cost reduction because we have to provide access to medicine to all. And this has really driven me right now. So if you look at the production of mRNA by in vitro transcription, this is actually the different steps that we have to do from the plasmid production using the bacteria till the in vitro transcription using the different enzymes, DNAs, polymerase, but also capping, if it's not, if a poly-A is not encoded in the plasmid DNA, you have to add a poly-A polymerase. So of course, there is a very huge benefit on using this. It's a reduced time and the manufacturing footprint. There is also flexibility, easy manufacture without a significant change of production process. But I have to say, some of the supply chain is a little bit expensive, especially in GMP grade, because of the proprietary components. It's a high cost. So there are different types of cost cutting strategy that has been proposed by different labs in the actually. So the short mRNA can be produced using a synthetic DNA template. Then you don't need to have a plasmid DNA produced in bacteria. And these are actually colleagues from Belgium is doing that. And you can also use a factory-in-a-box approach in which you have an automation production technology for the synthesis and purification. And you do it in bulk and you have a recirculations of the reagent. And of course, by doing so, you have a reduced cost. The other way to do is a bioproduction in cell factory. And I have to say that in 2001, in the beginning, when I started to propose and to do this, I didn't really hear too much about, this in addition to us. But now there are other people who are actually doing proposing this bioproduction in cell factory. So of course it's becoming there is a competition, but in a way, maybe it's validating what we are doing. So I applied the grant, which is called the EIC Pathfinder Challenge Grant, High Risk, High Gain, which is called YSCRIP for bioproduction in East. So I already told you, why do we have to reduce the cost? If you would like to do this, you need to actually have a cheaper source of reagent. You can also, there is some company who is actually producing ribonucleoside from bacteria or yeast, and then using them for the IVT. And of course, it's reducing the cost. But you can also switch in vitro transcription to in vivo production. So if you want to do that, you have to find the right host cell as factory, and you have to get these structures of mRNA, but is known to be very efficient right now with CAP1, modified nucleoside, as proposed actually as the milestone work by Professor Carico, Andrew Weissman, and Professor Weissman, and of course poly-A-TAL. You need also to master the challenge of in-cellular production, which is not so easy. So at the beginning, you have to think about what type of cell factory can I select? You know, if you look at the bioproductions of protein, you know that you can use bacteria. Bacteria as a low cost, genetically programmable producer with, and you can use it, you know, for bioactive natural product. But if you look their mRNA metabolism, it's not very similar to what you have. So you need to make a very tedious manufacturing. And of course, there is a safety concern about bacteria. You will, you know, end up to exactly the same limitation as for the in vitro production using the template from bacteria. You can also use mammalian cell, which is a perfect host, right? But there is a high cost of cultivation. It's a complex engineering. Mind you, maybe now you can do it easily. And the mRNA process is hard to handle. So if you look at just want to, because there are a lot of students in the room, if you look at this is the kind of cartoon of a different process that you can find inside the cell. It's already a little bit old because it's 2022. You can actually use a cytosolic expression to avoid the limitations of nuclear import. Here, you express the T7 RNA polymerase actually in the cytosol. And in this way, you can get your mRNA directly in a cytosol. The question is, are these mRNA will be in a very good shape, everything will be okay? How can we handle the accumulations of the mRNA? Is it going to be translated as soon as it's expressed? And if you do it if you look at the intracellular routine trafficking of mRNA inside the body, we know that there is a storage in the P bodies, storage in the stress granules, storage in maybe another types of granules that we don't know yet, storage in a condensate. I think if you look at this very complicated cartoon, it's quite difficult to handle this. In addition, there is a cost of culture for mammalian cells. So we end up to the yeast. Yeast can actually resolve different issue related to low heterologous pharmaceutical product yield in E. coli and in mammalian system. Why? Because it offers less susceptibility to contamination by phage, and you have an improved secretion efficiency than bacteria, and it does not contain endotoxin. Of course, we are not going to secrete, at least right now, our mRNA. But still, I'm just giving you all of a very positive issue with the yeast. Yeast is known as grass, as having a grass status, meaning generally recognized as safe. And it's capable to produce several heterologous and pharmaceutical proteins, bioactive molecules. And I think one of the most important thing with the yeast is the fact that yeast RNA metabolism is really transposable to mammalian cells, because we have most of the exactly the same or similar process that we have in mammalian cells. You can find exactly the same in yeast and in mammalian cells. Of course, it's not the same proteins, but at least they have exactly the same function. Today, yeast can be used to produce cytokine, blood product, hormone, enzyme, antibodies, vaccine. And you can actually, transform the yeast to 1, 5, many genes to get those different types of proteins. So the idea is, could we produce and accumulate mRNA coding for those different types of therapeutical product or vaccine? But instead of producing the mRNA, the protein, we have to accumulate the mRNA. So this is the goal of the YScript. It's productions of bioproduction platform from large scale for large scale and cost-effective manufacturing of mRNA therapeutics. This is the ultimate goal, I would say. The vision is to design a tailor-made yeast-based mRNA factory platform, which is, as you know, a complete shift of paradigm compared to IVT production. And it's a new use of yeast as a cell factory. So if you would like to do that, you have to get a transcription of a template DNA encoding a gene of interest in a capped and polyadenylated mRNA, similar to the mature mRNA in human cell. You need to have an accumulation of mRNA in a compartment or a forms that avoid or reduce degradation and translation. And of course, which is really the graal, I would say, you have to get a high grade of purity. So here, just to have a very simple 4 goals for us: produce engineered yeast strain able to accumulate mRNA of gene of interest, develop a robust and scalable yeast bioprocess for mRNA production, and of course, design an innovative and cost-effective process for mRNA extraction and purification from yeast, and make a proof of concept that it's working. So of course, it's very, very difficult. It's very, very challenging, I have to say. The idea is to have something like at least 10 or 50-fold less in terms of cost. You can do it in 2 ways. Either you can have a very high production, or you can produce something which is highly, highly active, okay? So the YScript consortium is actually composed of many partners. The first partner is Tron. Tron is actually the institute that has been created by Ugur Sahin of BioNTech. It's a translation and scientific organization, and they are very expert on mRNA platform, conventional and auto-amplifying mRNA. Of course, you have I was at the CNRS before. Now I am in INSERM, but at the University of Orléans. INRAE from Toulouse are working on bioprocess pre-industrial integrator in Toulouse, in France. And then we have Aveiro University and, of course, Taberia Interior University, Fanny and Mara Freire for extraction, clarification, and purification. And IBCH PAN, they are biophysicists working on mRNA complex of chaperone and mRNA interaction, and of Sartorius bioseparation from Slovenia. They are very well known in purification and quality control of mRNA, and the management is actually handled by EURIS, which is they are in Germany. So all of us different partners are working on generation of this cell factory, integrated extraction and purification, bioprocess development, quality control, and of course, all of this is actually by myself and with different types of, with different colleagues. So I would like to show you, as I said, I apologize, I cannot say everything, but because you can understand it's protected. But I think it's interesting to really give you how we actually dissect and trying to build this factory. So from the yeast cell engineering, the idea was we need to express in a simple idea, in a very simple way, I would say like that. We need to express inside the yeast, to transform the yeast with a plasmid coding for the chaperone. There are different types of chaperone that are able to recognize the mRNA. And in another plasmid, we have an mRNA of gene of interest. The idea is to have this mRNA of gene of interest fused with addressing sequence that can be recognized by the chaperone. The chaperone can be one protein, can be two protein. You can actually make different mix of it. Then once you have this, you need to find out, because the chaperone is recognizing the mRNA. If you look at the chaperone protein in the literature, the addressing sequence sometimes is huge. It's conformational. It's not, you don't know it exactly. So you need to understand, so you need to cut, you need to find out, you need to fuse. So this is what we've done at the first place, trying to see which part of the mRNA could be recognized by the chaperone protein. So I'm just showing you here. So everything, the plasmid DNA is the coding sequence is under GAL1 promoter to get it unusable with galactose. So here is just a confocal microscopy showing you that if you express your reporter plus alone in the yeast, you can get very nicely your expressions actually of your chaperone. This is the reporter. You don't see it very well. If you have your reporter plus the total amount of the chaperone that can recognize also its own mRNA, you see a very, very small colocalization because it's an endogenous recognition. If you try to really find out the best addressing sequence from the chaperone, you see that it's starting to have a very huge condensate formed by the protein plus the mRNA. And depending on the regions of the chaperone, you can find out huge condensate, sparse condensate. One of the best image that we can have, I am showing you here, is actually a very well colocalization between the chaperone and EGFP mRNA that we have used to be recognized by the chaperone. So we have found out that 88% of the mRNA in these very well best cases are actually colocalized with the chaperone protein. So when we wanted to, so it shows that somehow we would have an accumulations of our mRNA inside the cells. We need to really validate this. So what we did was we use our chaperone, we fuse it with EGFP, allowing us to follow as a function of time the percentage of cells, yeast cell expressing the GFP. And we also transform the cell with luciferase plasmid DNA plus the addressing sequence recognizing the chaperone. And then if you look at this flow cytometry, you see that at the beginning, before the induction, as soon as we started the induction, after the induction, we have a high percentage of cells, almost 50% in this case, which are actually transformed with the EGFP, okay? And if you look in the population, we have some of the cells are very highly expressed, GFP, which is in blue here, after 2 hours, and then it's stable. Some of the cells are very low expression of the GFP, and the other one, I would say, in the middle. But here it shows that at the beginning, we have a kind of not an homogeneous, because it's not a cloned cells, of course. And if you look at the MFI, the amount of chaperone GFP that is produced over the time, you see here, but you have an increase as function of the time. It shows that indeed, maybe if this chaperone is recognized in the luciferase mRNA, we could have this accumulation. And then we took those cells and we actually extract the RNA and we try make an RT-qPCR to see the amount of luciferase mRNA inside the total RNA that we have been extracted. And here again, you show here, as a function of the induction time, we have an increase of the mRNA, and see here, at 15 hours, we have a high amount of this mRNA. And here it's showing you the accumulation. If you look at the chaperone mRNA, it's exactly having the same trend. But hopefully for us, actually, unfortunately, the this is one of the big issues that we have, the luciferase mRNA and the chaperone mRNA is a little bit, it's not having the same amount. So we are working on this now to reduce this amount of the chaperone of the mRNA from the chaperone protein. Okay, and here, if you took this RNA and do a in vitro translation, because it's coding for luciferase, what you see here is that as soon as you induce, this one is just the I'm sorry, I make a miss. As soon as you transform the cell, if you luciferase mRNA, it's very high, the RNA, and then it's decreasing. But for the strain expressing both the chaperone protein and the luciferase with the mini addressing sequence, you do have an increase, and you see here that up to 2 hours, the expression, the RNA, the mRNA inside the total RNA are actually able to be translated. I'm going to re-explain this one. So here, the strain transformed with luciferase only. You have also an accumulation, but it's very less than that for one that having actually the chaperone and the luciferase. So increased accumulation of luciferase mRNA, very low translation of the mRNA luciferase protein from 2 hours, okay? And it's why? I think it's normal, because when we do the extraction, most of your mRNA is actually in a condensate, and it's not able to be expressed as a luciferase protein by the cell. Then we took those cells and we look if indeed we can actually find out the mRNA of the coding for the luciferase inside the cells and the anti-chaperone Cy-free. And actually what we found was there is a 80% colocalizations of our chaperone and the mRNA of the luciferase, which has been detected by sheep. So here is by fish. So this is actually showing you the accumulation, the condensation, and because of this condensation, there is no translation. Now, it's very important to see that there is a stability of this mRNA inside the chaperone condensate. So here, we made this kind of experiment. We started to make a pre-cultures of our yeast strain, then we induce it with the galactose, and then we stop it. We stop it with the glucose, so it allows us to see all of the proteins that has been expressed, or the mRNA that has been expressed, how it actually evolves after the blockade of the galactose promoter. So here shows you that it's an RT-qPCR, just 15 hours after the induction, you see high amount of mRNA in the strain that has been transformed with both GFP, chaperone, and luciferase, whilst the one having a luciferase alone, it's very low. And you see also that indeed, after 25 minutes, there is a decrease, but this decrease is a little bit stable. You see here, up to 17 minutes. Then it started to reduce, and it's it's very significant. It's significant from here to here, and also from here to here. So it allows us to really get the the window time, the time windows where we have to work on. And if you took those RNA, it's not mRNA. Just take the RNA from those cells, and then we make extract from the condensate, and then make an in vitro translation. You see here again that indeed we do have expression of our RNA. And of course, because here there was a reduction, you still have RNA that is intact, but it's a little bit less than this one, indicating that we need to have, it's not a little bit less, it seems to be a little bit less, sorry, but actually it's non-significant. So it shows that all of the RNA that has been accumulated inside the condensate actually are intact, and you can translate it in vitro. And again, I said, it's not mRNA, it's just the total RNA that you have used. We took those condensate and we tried to evaluate how is the size, how it looks like. At least in our hands, we find out that we have something around 150 nanometer by video drop imaging. And by SPR AFM, we also found a 2.5 10 to the 9 particle per ml from our extraction. You can tell me, it does not mean anything because I don't give you the volume at the beginning, but I cannot give you this. But indeed, what I would like to say is that we do have approximately the same amount of particles per ml. We took this mRNA, we took the RNA, and we tried to extract the mRNA. So we had a very huge problem here because at the beginning, we found out a very good ribodepletion kit. And sometimes, from time to time, the ribodepletion kit is not very well. Anyway, when we started to get, at the beginning, when you had a very good kit, we were actually able to assess the length of the poly-A from our RNA production, and it's around 120. And I just want to say that we have added 120 poly-A tail in our mRNA on plasmid DNA template. So this is actually, we expected, okay? And to do that, we have used what is called a poly-A tail length assay of Antimetrics. And now we are trying to validate it using another types of a method with mass spec or another types of kit, just to make a very good validation. So then we also give the RNA and send it to Fasteris company to make a next generation sequencing to see if actually the coding sequence that we are expecting to have have exactly the same structure, the same sequences than the theoretical sequences that we must have. And we are very happy to see that with two different types of mRNA of gene of interest, we obtained transcript length, which is 99.9% identical to the theoretical sequence with a mean of 30,000 reads per base. So it shows that there is a kind of fidelity of the transcription that has been made from the plasmid DNA that we've used to transform the yeast. Now, one of the big, I think, great issue, great thing, sorry, about the yeast is the fact that they are also making modifications of mRNA. They are also having an epitranscriptomic process. And it's very important to say that there is a kind of conservation of these modifications of the mRNA. So it means that somehow we can have an endogenous modifications of our mRNA when you produce it in yeast. So our first experiment to really show that from the mRNA that we extracted, if you can find a modified nucleoside, is actually these series of experiments that I am showing you here. It's a stable isotope labeling coupled mass spectrometry. It has been done with Stephanie Kellner, which is in Germany, in Munich. And actually, she is using this process where she cultivates the yeast with a specific medium, and then she can we can actually digest a nucleotide, and by making a comparison, I don't have time to show this, she knows exactly where is the abundance of modified nucleoside. So here we use the luciferase RNA, and actually from the RNA purified condensate that we sent to her, she found out that there is different types of modification from the mRNA structure, which, by the way, some modification that we can find in our mRNA. Then we decided to do also a ribomed-seq analysis, and this one has been done in Nancy, RNA-seq platform, and here, first RNA structure, they make positions of 2' methyl modified bases. And you see here in all of these little red dots is indicating the methyl base, 2' methyl base that they found. And then this is one first mRNA. And then I said, maybe we can actually have another mRNA. But instead of having just one, we can make a 2-3 batch of bioproduction to see if we can have exactly the same place, the same position of the different modifications. And this is, you can really see here, actually from the 3 runs, he got exactly the same position, indicating again, but there is a fidelity of these modifications of mRNA. Yeah. But the main problem here is the purification challenge you can imagine, because you know that most 90% of our RNA inside the eukaryotic cells are actually ribosomal RNA. Here we have used RNA coding for luciferase. And we are a little bit unlucky because we do have this RNA from luciferase is exactly the same length as the 18S RNA. We've done a lot of English IVT LeucMRNA, so I'm just showing you here. But now, of course, we are switching to another one. And you see, there is a very huge contamination. So this is the IVT. This is the IVT. This is the poly-A RNA from the condensate. So, of course, it's a little bit less. When I said poly-A, it means that we make a purification with oligo-DT here. And then if you make a ribodepletion, you still have exactly the same thing, a little bit less, but still the same thing. And the 5/5 one is actually the total RNA, which is a huge amount of this ribosomal RNA. Still, if you take this and you make another blot, you still get exactly, you still find out that here, indeed, there is the luciferase RNA here. And it's depending on the amount, on the purification, because here in the 5th 10 microgram of our total RNA, of course it's there, but it's very, very small compared to this one. So high contamination. So if you have something which is working from this contaminated mRNA, it means that it must be highly efficient. Anyway, we also make a design of innovative and cost-effective process for mRNA extraction. For that, this is a work from University of Aveiro and University Beira Interiors, I mean, Fanny's and Mara. And actually, they really make a very mixture of different ionic liquid synthesis to actually lyse the cells Then we use it with different combinations of polymer to make a concentration. And then Fanny is coming with the chromatography purification, where she's actually adding some amino acid to recognize, and also some specific support, colon support. I cannot really disclose everything, but I just want to show that indeed, from the lysis, this is the classical lysis, using beads, using, to lyse the cells plus some enzymes sometimes, you can have a very high, very good extraction, shown here by this very high amount of ribosomal RNA. But if you use 2 and 3 specific bio-based ionic liquids, what is important to show is that indeed we do have extraction, and the amount of those ribosomal RNA is less. So clearly it's indicating that we do have something which is interesting that must be optimized to get a very good process. Here it's a luciferase translation, luciferase assay, in vitro translation. So before the process, you have 10 to the 5 luciferase unit, and after the process, we have 100-fold more, indicating that indeed we have reduced the contamination. Pani is also, as I said, modifying this, and this is done with Biosartorius, bioseparation Sartorius, and now they are actually optimizing the process, and I cannot disclose. But of course, it shows that the accumulation is there, but maybe it's not optimal. So what we've done to really improve this recognition, so we made a systemic evolution of ligand by exponential arrangement. It's called SILEX. This is done in collaboration with Institute of Biophysics in Poznan and with Tron. And what they've done was they make SELEX. SELEX is a evolution arrangement, I would say exponential arrangement, where you actually take a random library, you cut it, library that could be recognized, of course, by the chaperone protein. Then you make the binding, you wash it, and then you elute, make an RT-PCR, and then do different round, 100 rounds. And in the end, actually, you take them, you modify your plasmid DNA, and you see if there is an accumulation. So I'm just showing you that indeed, Kasia Pasurska, they find out one very good, aptamer, because it's called aptamer, because it's a short length of oligonucleotides, RND-A1 and RND-A2. Those, this one and this one, those two are actually able to, I would say, compete with the mRNA that can be recognized by the chaperone. So we took those aptamer and we chose this one for different reasons. And here you see, instead of having the luciferase and the mini addressing sequence that we have before, we had this in addition to the mini addressing sequence, or we had this alone, or we had double it just to see what happens. So here we have, indeed, we found out that when we make the transformations of the yeast with those different types of luciferase, with those different types of addressing sequence, clearly, if you take the RNA from those yeast, it's a total RNA. I really want to say that. And we look at the in vitro translation, we found out that indeed this one, even though we have a very high deviation, standard deviation, this one seems to tend to have a better, I would say, activity compared to this one, which is the classical one. By the way, R means for condensate, S means for supernatant, which is not the cytosol of the cell. So you see that everything from the cytosol does work because everything is accumulated. And if you look at strain by strain, this other presentation of this one, indeed, there is still now, we do have a kind of heterogeneity. So we are trying to actually finding the better clone to get this clone here. But clearly, we do have this one, which is better in terms of efficiency compared to the first one. So this is still ongoing. But I told you also, but we need to have an mRNA bearing CAP1. CAP1, I'm not going to take a long time, but to explain to this, but you know that CAP1 is the 2' ometyl, required to have a 2' ometyl transferase. It's actually distinguishing the mRNA from prokaryotes to the mRNA from mammalian cell, but yeast cell have a CAP0 as the prokaryote because it's a lower karyotic cells. So we need to have this CAP1 because if you don't have the CAP1, you have activations of a guy, you have activations of innate immune sensors, and this is actually inducing inflammation, inducing degradation, in addition to the pseudouridine modifications, as is shown by Professor Carico and Professor Weissman. So we made modifications of our yeast. I'm not going to disclose this. I'm really sorry, but I just want to show you the data from this. Again, to see the accumulation, you did exactly the same methods that I just shown before. And here is actually the luciferase expressions of the mRNA. If you take cells transformed by luciferase only, luciferase plus the chaperone, luciferase plus the chaperone plus the CAP1 process, you see here that indeed luciferase only is very low in terms of expression. And here we do have high expression with the chaperone. This one is with the one having the CAP1. And if you look at the function of time, I think it's very clear that there is a gain of stability compared to this one. It's almost a doubling. And of course, from 40 minutes, it's a bit, it's lower, but we have work to do here to really improve this and either stabilizing it or increasing it to this one, to this. Anyway, despite this data, we decided to see if RNA from those different strains can be actually used to express the HeLa, to be used for transfection of HeLa cells. Again, here, if you look, control cells in vitro translation, this is the one without the CAP1, this is the one with the CAP1. So we have an increase. It's a kind of two-fold, 2.5 fold. And if you use this to transfect the HeLa cells with lipofectamine, you see exactly the same thing. This is the mRNA without the CAP1. This is mRNA with the CAP1. Again, we do have exactly the same thing. I just want to say that in the literature, and even in my lab, if I do have a CAP0 versus CAP1 transfection efficiency, I do have at the maximum 3-fold more expressions of the protein. This is what we have here. So now we have improvement of the luciferase activity in favor of the presence of CAP1. We validated by mass spectrometry. I don't want to show it because I have to repeat the experiment. Of course, it's there, but yeah, something is actually, we are actually, building all of the bricks, and I really hope that we will succeed in a few months. Last, of course, all of this is great, but we need to have a robust and scalable yeast bioprocess for mRNA production. It means that we need to optimize the yeast culture condition. We need to have the scale up. So for that, we selected the best factory. we make optimizing the experimental condition. And of course, we evaluate the genetic drift. And in the end, if everything is okay, you make a pilot scale process validated for industrial development. Very quickly, the growth kinetics, 3 different types of strain. I think it's quite clear here, but this strain 3 is better than the 2 and then the 1. And if you put the chaperone is making the condensate with GFP, you can follow the formation of a condensate with a GFP positive cells. And again, you see here the shift is higher compared to this first one before the induction. Okay, so the third, we have a strain that is working very, very well in terms of culture. Now, if you are doing bioprocess with the yeast, we have to follow up the different metabolites that you have. So here is just a curve allowing you to show how is the consumptions of the glucose, the consumptions of galactose, but also the expression of the GFP and EOD, I mean the culture, the biomass. So from this strain 3, you see that before the galactose, there is a consumptions of the glucose. When you add the glucose, of course, you have a stable consumption of glucose, and then it's going down. And at the same time, you have an increase of the OD, and you also have an increase of the GFP. So what is very important is actually here, the ethanol is actually reducing after 15 hours. By the way, if you remember, I showed you that the maximum of our accumulation in my previous slide was 15 hours. We see it exactly in this curve of OD and metabolites from this strain. So yeah, so actually many things are going on. I hope that we can actually have a process scale, which is industrial scale, that will be validated. But just to summarize, and the next step, we actually able to accumulate our mRNA of interest with endogenous nucleoside modification. We have a correct poly-A tail. We have expression in vitro and in vivo. Of course, we do not have a purification, highly purified, but still, your transcript rate is 99.9% identical to the theoretical sequences. And we can actually engineer the yeast with the CAP1, and this mRNA from this yeast have a 2.5 and 3-fold efficient in terms of efficiency as for IVT mRNA, if you compare CAP0 versus CAP1. The challenge, I told you already, is extraction. We still working on the purification and also on how to have a very, make a lysate with a very low, without using any harsh condition. We need to evaluate the critical attribute and key performance indicator and cost effectiveness. I just want to finish by saying something. If you do a bioprocess, what we have to think about the quality aspect of the mRNA. Today, EMA has only a quality aspect of mRNA vaccine because the field is still growing, and there are a lot of big people who are working on what regulation we have to put. I just want to show you that if you look at the principle of the ICH Q6B guideline for mRNA vaccine, it shows you need to have an integrity of primary structure, 5' cap, UTR, ORF, 3' poly-A tail. You have to do this either HTS, high throughput sequencing, NGS, or oligomapping. We have to have to know the order of a high order of mRNA structure. There are different ways to do this. We are doing a shape to say what happened, what we can have. Do we have exactly more or less the same as what we have in vitro? And I think it's very important. The product-related impurity, this is really, really, really mandatory. What do we have? Do we have a different size RNA once we make a purification? Do we have a nucleotide modification? Do we have a 5' or 3' poly-A tail variant? And of course, the double-stranded RNA. Right now, from the yeast, we do not have a very high amount of double-stranded RNA. In my lab, sometimes you do not have any. And of course, the functionality. And I just want to finish by showing that, yes, as Katalin Carico said, we need to have a company to try to take it and actually to work on it. I'm very, very glad that bioRNA is actually two of my colleagues, Pascal Viguier and Thierry Ziegler, actually believe in the technology, put the money on it, and we are working to de-risk the technology right now. So we hope that we will raise more money to take it, to get the pilot scale here, and actually to have the large scale production. Yeah, and this is really the very, very high challenge that we have. So if you would like to get this mRNA clinical quality control, it means that we need to have to know what type of fragment we have in terms of DNA. We need to know if there is actually residual, maybe we don't know, host cell protein that could be there. We're starting to do it already in the lab. It has to be very, very low, 5 nanogram per mg. I hope that we won't see any. And this one is also going on in the lab to demonstrate that there is no beta glucans left during the lysis, because this is very, very inflammatory. From that, I would like to thank my fabulous colleagues. It's a tough project, it's a challenging project, but yeah, this one was in Slovenia, this one is here in Tron, and yeah, and I would like to thank you for your attention, and thank you so much, Fanny and Bruno. Thank you.
Thank you so much, Chantal, for the excellent talk and for all the scientific insights that you shared with us. And now we have some time for questions from the audience.
You know, if I can ask a question, Chantal,
Yes.
so that the nucleoside modification, I noticed that the methylation on the N1 on adenosine was quite high, and that would interfere with hydrogen bonding. So it means that the tRNA, you know, when it translation, it won't translate. And 2'-methylation is a good thing because it makes the RNA more stable, and we cannot introduce that. But in that case, also in some tRNA interacting with the 2' hydroxyl on the ribose, so that also could interfere depending on. So that I don't know whether what specific enzyme is methylating, for example, the adenosine on the N1 position, whether it is special and it can be knocked out, so
Yes.
that it won't do, or I don't know whether it is lethal would be, or,
Yeah.
you know. But yeah, I mean, just all of this work, I mean, just amazed. I was listening here how many things you have to attack on it, and it is great. And I kept one, probably you can take in the enzyme, you know, knock it in the enzyme, I don't know, in the yeast.
Actually, we managed to do that, Kathy. It's working. Actually, it's working. Actually, I managed to. There are different ways to do it.
Yeah.
Of course, as you know, I can tell you maybe in a confidential way. I'm not going to say.
Yeah. That's fine.
But indeed, yes, we managed actually. It was not easy, right? Because it was not easy. But we were able to actually find out a way to modify the yeast that it allows to add, add the 2'-O-methyls on the cap zero. So this is okay. I fully agree with you with the modification. The question I have right now is that I know that there are some modifications that could not be good, I would say, for the translation. I know also that some of the modification could really be very, very interesting in terms of stability and in terms of avoiding maybe some, I don't know, INET sensor or whatever. right now, our goal is really to show that when you made the production, when you took it, when you made the modification, we are actually able to express
Yeah,
a
that you
protein
could translate.
inside the cell.
Mhm.
And at the beginning, I must say, I was like, oh my God, this is not gonna work. But indeed, despite the purification, which is not 100%, I would say, which is just even 70%, even maybe there is still 30% of impurities, and we do have something, which allows me to say that we do have something which is very efficient.
And listen, how much is the cost if you are doing with yeast versus when you would do with all of these capping analog enzymes and so on?
Uh,
Yep.
Actually,
Your estimate, the best estimate that if
Oh,
everything works out for you, the purification, everything, what do you
our
think?
best estimation right now is maybe I would say tenfold less.
Mhm.
Jeremy is actually in the room. I saw it. It's tenfold less right now. If everything would be optimal,
Yeah.
we really think that it could be 50-fold less
Mhm.
if everything is optimal.
Mhm.
But again, maybe the main, compared to, I would say compared to 2.5 years,
Mhm.
I think we gathered a lot of knowledge,
Mhm.
and we are, so I really hope that having a company, because you need to have a company, because they are the way of their thinking. So the follow-up of the Y script now is another European project, I hope, for a company.
Yeah. Congratulations.
Thank-
I should start that it was great. I just listen and amazed all of this. Thank you.
Thank you so much, Katie. Thank you for, I mean, I would never, I mean, I would never do it if you told me, oh, Chantal, what are you doing? I would say, I would never, because, it's all, I am one of those, and this is also I learned from you. Try to have a little bit of self-confidence, because I don't, it's maybe because, and this is just to tell everyone, if you are a woman, and you are, I don't like to complain, because it's not a complaint. It's not good to complain, but we have to say that this is a world that has been made by men, right? And sometimes it's a little bit difficult, but again, I'm very happy. I mean, the INSERM asked me to give something, and I'm doing something that I like. I'm doing something that I built, and I think it's great. And I met great people as you, as Drew, as a lot of people. So thank you so much.
Thank you. Maybe I can also make a question, Chantal.
Yes.
Well, despite I'm involved in the project, but maybe I will have a question for all the audience. Okay, is this technology robust enough for different kinds of messenger RNAs? What do you think that research can lead us?
Yeah. Yes. I mean, we used conventional mRNA, but of course we can use it for. Actually, the patent is for different types of format of mRNA, of course, and we also already produce long length mRNA, short length mRNA. So yes, it's working.
Okay. Thank you. Yeah, we have also a question here in the room.
Hello. Hi. Thank you so much for the great presentation, Professor Chantal. I was wondering, since you now want to go to a large scale, what is the main issue with the regulatory agencies for it to be applied? Would it be that, and also in the whole process, would you say that the most difficult part is to obtain a high quantity of RNA with that production, or to purify it very well to be then administrated. So
You know what, the good thing with the yeast, thank you for your question. The very good thing with the yeast is that there is a lot of infrastructure of yeast production right now. You can have 300 liter, we can have 300 liter multiplied by 10, you can have, this is already, it does exist, right? So you can do this. Of course, we need to improve the amount of mRNA that we have to produce. I'm convinced that there is a different way to even improve it. For the regulatory, we already discussed with them, at least with the French one, and I think I would say we are lucky in a way that we know exactly already what happened, what are actually regulation for productions of protein, okay? And we know also for the in vitro mRNA. So now we need to build, I would say build another track with taking the knowledge from the in productions of proteins from the yeast. So, you know, not to have host protein, not to genomic DNA from yeast, all of these things, and also having a very high purity. So I think this is so it's not easy. I don't say that it's easy, but it's I think it's feasible.
yes, it's challenging. Very interesting. Thank you so much.
Okay.
More questions? Yeah, maybe the last one because we are a little bit late in our schedule.
Hi, great presentation. So I work with microRNAs, non-coding RNAs. So my question is, why messenger RNA, and what is the advantages of these RNAs against non-coding RNAs? Or both can be used for different things?
Oh, you should ask this to Katie Carico, actually. You mean, listen, you mean for me, why do I produce in mRNA instead of microRNA?
Why the choice of mRNA?
Oh, listen, you know what? I mean, I've started, the choice of mRNA for me is it's allowing me to produce everything. It's allowing me, it's a very flexible modality. It allows you to produce either for vaccine, for different therapeutic protein, cytokine, hormones, proteins to modify the microenvironment of the tumors, replacing something that is not there, or, you know, expressing something that's not there from autoimmune disease. It allows you a variety. It's a very high flexibility. MicroRNA is also, it's great, but by the way, you can also maybe produce a long non-coding RNA with this kind of production. It's possible. If this is a question you are asking me, yeah.
Thank you.
And by the way, I think it's in a patent as well.
Thank you, Chantal. This was Micaela Riscado that is actually working with microRNA. So I think this is the interest in the question.
I knew it, but it was Micaela. I knew it.
So, and maybe just one last comment. So YScript project is finishing, which means that the funding is finishing, but the project, the ideas and research will continue, isn't it?
Yes. The idea and research will continue, and that's why I'm very glad that bioRNA is taking it.
And of course, we have to expect good results and the translation to of this technology.
Yeah.
So thank you so much. So let's thank again our plenary speakers.
Thank you. You can say the thank you like this.
So actually, I have to express my deepest gratitude to the plenary speakers, Professor Kathleen Carico and Professor Chantal Pichon, for their inspiring talks, for the invaluable contribution to this workshop. It was really great. It was an honor for me to host this and to have this discussion, to learn from you. So thank you so much. And before moving on, and because we will leave the transmission, the online transmission, I have just to thank to all my team, my PhD students. We played a key role in organizing the workshop. So I'm here, but they are all in the backstage controlling everything. So thanks Bruno Batista, Rita Carapito, Pedro Ferreira, Micaela Riscado, Marco Luís, and Tiago Graça. Also thank to Dr. Rui Costa, who helped us with the informatic questions and to put this online for everyone to assist. We had a lot of persons attending and by Zoom meeting, and also following the transmission on the YouTube. So thanks to all who have been here in the room, and thanks all the participants also who are attending online. So thanks for joining us and being part of this event. And now, as I said, with the conclusion of the plenary sessions, the online transmission will end, but we will have a coffee break and we can continue to our next part of the program, the RNA Uncovered, an end-to-end scientific tour, where the on-site participants will have the opportunity to explore the RNA biomanufacturing process and all the advances that the University of Belém Interior and the University of Aveiro have made related with the YScript project. Thank you so much. Thank you all. Good
Thank you
afternoon.
so much. Thank you, Fanny. See you soon. Bye-bye. Bye-bye, Katy. Bye.